primer extension or the single primer method, it is another method that is also a simple one for site directed mutagenesis. इसमें हम एक प्राइमर को यूज़ करते हैं जिसमें हमारे चॉइस की म्यूटेशन होती है, और ये जो प्राइमर है इट मे बी अराउंड 7 to 20 बेस पेयर लॉन्ग्स, और अगर इस प्राइमर में एक या दो बेसिस जो हैं ये ऑल्टर्ड हैं, म्यूटेटेड हैं, हमारी चॉइस की हैं, तो ये एनीलिंग को प्रिवेंट नहीं करेंगी. At least annealing is there because of relatively short stretch of the primer. And after annealing, the in vitro DNA synthesis it is carried out, so that the bases that are mismatched as compared to complementary DNA or the target DNA, it is introduced within the complementary DNA. कि जो प्राइमर में जो मिसमैच बेस है ये हमारे टारगेट डीएनए में इंट्रोड्यूस हो सकती है. ऑल द फ्रीक्वेंसी जो है ये कम या ज़्यादा हो सकती है. सो फॉर द पर्पस टू इंट्रोड्यूस mismatch bases or mutated bases in the target DNA, it is important that the target DNA it may be available in single-stranded form. So that within single-stranded DNA, the primer it can be annealed that contain one or two mismatched bases. इस पर्पस के लिए अगर आपको याद हो M13 वेक्टर जो कि single-stranded DNA होते हैं, it can be used. But in addition to that, plasmid DNA which is double-stranded, it can be converted into partial single-stranded form. It is equally suitable. Now, what is the basic strategy in the primer extension procedure? Here we can compare first two methods. The first one I explained related to cassette mutagenesis. Another approach of site-directed mutagenesis, it is of primer extension procedure. So here we have single-stranded recombinant DNA or vector, and having the sequence of the gene suppose CAG. Now we want to replace A adenine with another base. Suppose here it is C rather than T during the process of DNA synthesis. And here it is the cytosine is there in the center that we want to target. Now after annealing, the DNA synthesis it will be carried out. So the double-stranded DNA it is produced. Now this recombinant vector after primer directed DNA replication, it is used to transform host organism, and after transformation some of the progeny cells they will maintain the wild type genotype, that is normal sequence it is restored by the repair machinery of the host, whereas there may be some frequency of the mutants or progeny cells where now the mismatch based in the center it has been replaced. It can be explained with the help of another diagram. Here an oligonucleotide or primer directed mutagenesis it has also been explained. For example, here M13 vector recombinant vector is there having a particular sequence of the gene that is mentioned here, and here the central one adenine we want to change, and that is during the DNA replication instead of thymine in the opposite strand we want to introduce another base like suppose cytosine or guanine. So for this purpose we have chemically synthesized primer or oligonucleotide where the G is present instead of A. So after annealing, the DNA replication is carried out in the presence of DNA polymerase and 4D NTPs and T4 DNA ligase and ATP it is also required for this process. So it will direct the synthesis of second strand, and annealing it is not prevented because of just one mismatch is there, and it can be used to transform E. coli cells. And after transformation, what may be the possibility in next step? One of the possibilities that most of the progeny cell they will restore the wild type phenotype, because of the host machinery, but in some cases, in place of adenine, the cytosine is there. So it is misprinting here. It is actually cytosine. So guanine will introduce mismatch, that is cytosine, and it is later on screened. After transformation of the host organisms like the E. coli, the progeny cells, they will divide, and two type of heteroduplex may be produced. So some of the progeny cells after division, they will retain the wild type gene sequence, whereas some progeny cell, they may contain mutated base or mismatched base. It is incorporated, although the frequency may be very low, but such type of mutants are there, and those mutants then they are detected. And for this purpose we must have a probe, a short DNA sequence, you are well familiar, that can be used during the process of hybridization. और इस पर्पस के लिए जो stringency control है वो बड़ा ज़रूरी होता है कि the degree to which the hybridization probe it annealed or hybridized with the target sequence. It is also very important. So stringency can be controlled by controlling the temperature या cation की जो रेशियो है ड्यूरिंग हाइब्रिडाइजेशन प्रोसेस उसको भी कंट्रोल किया जा सकता है ताकि सिर्फ पॉजिटिव सिग्नल आए. Positive signal means that if our mutated gene or base has been incorporated, it must be detected. So it can be detected by the process of DNA hybridization by using sequence specific probe.